Hello and welcome to episode 640 of the People Behind the Science podcast. Today, we look forward to resharing our conversation with Dr. Lou Cantley, and this interview was originally published on April 11th of 2016. Listeners, Lou is a renowned cell biologist and biochemist who has made substantial contributions to advancing our understanding of cancer metabolism. His research group discovered and investigated the enzyme phosphoinositide, or PI, 3 kinase. This enzyme is now known to be important in both cancer and diabetes mellitus. And in our interview, Lou talks more about his work as well as his life. So listeners, get ready to be inspired by Lou's stories in this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. Listener, today I am thrilled to be here with Dr. Lou Cantley. So Lou, welcome to our show today. How are you?
I'm fine, Marie. Thank you.
Well, we are delighted to have you with us today. And I want to give our listeners just a little bit of background before we dive into our questions for you. So listeners, Lou is the director of the Meyer Cancer Center at Weill Cornell Medical College, as well as director of the Stanton Clinical Cancer Program at York-Presbyterian Hospital. He's also professor of cancer biology and medicine and professor in oncology research at Weill Cornell Medical College. In addition, Lou is co-founder of Petra Pharma and Agios Pharmaceuticals. He received his bachelor's in chemistry from West Virginia Wesleyan College and his Ph.D. in biophysical chemistry from Cornell University. Lou conducted postdoctoral research at Harvard University, and he served on the faculty at Harvard as well as Tufts University before joining the faculty at Cornell, where he is today. Now, Lou has received many international and national awards and honors during his career, including the 2005 Pezcoller Foundation and American Association for Cancer Research International Award for Cancer Research, the 2013 Breakthrough Prize in Life Sciences, the 2015 Ross Prize in Molecular Medicine, the 2015 Wolf Prize, the 2015 Association of American Cancer Institute's Distinguished Scientist Award, the 2015 Canadian Cairdner Award, and he was named one of the world's most influential scientific minds in 2015 by Thomson Reuter. Now, Lou is also a fellow of the American Academy of Arts and Sciences, as well as a member of the National Academy of Sciences and the Institute of Medicine of the National Academies. He's also a member of the European Life Sciences Academy. And listeners, Lou is here with us today to talk about his experiences in life and science. So Lou, we are delighted to chat with you today and to learn a little bit more about you as a scientist, but also as a person. So can you start by telling us a little bit about your life outside of science and what you like to do with your time?
I love doing science. So the vast majority of my time is just doing science and thinking about science and answering emails. But when I'm not doing all that, I love to read. I read quite widely. I particularly like to read science fiction, mystery stories, and also historical stories as well.
Very cool. Well, I know those emails can pile up quickly in your inbox, so I know it's a challenge just to keep up with those sometimes. But I'm curious to hear more about your reading. So you mentioned you like things like science fiction, mystery, some historical novels. I love to get book recommendations from everyone we have on our program. So Lou, can you give us a recommendation today of something you've read and really enjoyed?
Neal Stephenson. If you haven't read Neal Stephenson, he's a pleasure to read. He's a polyglot of science, and he writes incredibly entertaining stories that are fictional, but often based on some science facts or science history. The series of books I most enjoyed by him was something called The Baroque Cycle that was centered on the lifetime of Sir Isaac Newton, but most of the characters are fictional and incredibly funny and clever and sexy and sex and violence and all that in a background of science. It's really quite fun.
Well, we will put this out on our website for our listeners to find when they are in search of their next great book. And I know it's kind of hard to find time to do all the things that you want to do in a day. And you mentioned that you spend a lot of your time thinking about your work and about your science. And I know this is a major passion for many scientists. So can you tell us a little bit, maybe at a high level, about what your work is and how would you describe it to someone who's not as familiar with your field?
Well, the main thing that I do is study something that we call signal transduction. And I guess an analogy would be if you think about it, every cell in your body is a little microcomputer that has to respond to hormones and growth factors and whether or not it's contacting another cell. There's a wiring diagram inside the cell that allows it to make all those decisions. And basically, I've spent most of my career trying to unravel that wiring diagram and just understand at the molecular level how those kinds of decisions are made. Because virtually all of our diseases, the ones that certainly the diseases we can't currently readily cure by something like surgery, are diseases that involve defects in these signaling pathways. The cancer, for example, the cell is getting a signal to grow all the time, to divide, to make copies of itself. In other diseases, there's a failure of cells to grow. And these same networks are always involved.
Very interesting. Well, I'm excited to chat more about your research and some of the projects you have going on as we go through the interview. But I want to stop a moment and talk a little bit about motivation. I think you can sense your passion for the work that you do when you talk about it. And I think this passion is what motivates a lot of scientists. But I'm also one of these people who loves little motivational quotes and sayings, little things to get me through the tough times, because I know there are so many in science. So Lou, can you tell us, do you have a favorite motivational or inspirational quote that you use?
One of the quotes I like, I don't know if I'd call it motivational, but maybe it's a retrospective understanding of how things work, is a statement by Louis Pasteur that chance favors only the prepared mind. I find that almost every major breakthrough that I've made in my life as a scientist has come from an unexpected result. Often, for example, an impurity in a commercial reagent. So an experiment comes out completely different than what I thought it would come out. And I discover that all the results I've been previously getting were due to an impurity in the reagent. The reagent that I bought wasn't really what I thought it was. And it gives me a result that suddenly makes a breakthrough in how things work. So that sounds just pure coincidence, but things like this happen all the time and most people ignore them. I actually find that I learn from these mistakes.
So how do you keep your mind prepared, or how do you prime yourself to look at these things, not as like, oh, that was a mistake, run the experiment again, but wait, there might be something interesting here?
I'm always excited about finding the unexpected result. I'm not interested in dotting the I's and crossing the T's on ideas that have already been pretty fully developed. That has to be done, of course. There's always things that we can do better than we have in the past. But what's really exciting is when you break open a completely new field, when you realize something is working completely differently from anything in the textbooks. And the truth is that we understand so little about biology. There's some advantages to not even read the textbooks because they're just describing a very small fraction, a few percent of the complexity of what's really going on.
They definitely make it look way simpler than it is.
They have to in order for anyone to understand it. But the fact is there's a lot more to be discovered.
Certainly. Well, Lou, wonderful to hear a little bit about this mindset that you bring to science that helps you be so successful. I'm curious to hear about some of these people along your journey who might have contributed to teaching you or training you, or these people who served as role models to you. So can you tell us a little bit about them?
I'll name two people who've had big influence. My Ph.D. advisor, Gordon Hammes at Cornell, one of the most organized men I've ever met. He was trained as a physical chemist and kineticist, and he was interested in understanding how enzyme reactions are mediated, the structural and mechanical mechanisms by which these enzymes carry out reactions. So I learned a lot from him. I really started out as a physical chemist or biophysical chemist. And then my PhD mentor was Guido Guidotti at Harvard, who was an MD PhD. He had, of course, knew medicine, and on top of being an excellent scientist, an outstanding scientist. So I learned a lot about cells and membranes from him. And coincidentally, I just had dinner with him last night. I hadn't seen him in a few years, but it was great to see him.
Wow, a little reunion there. Very nice. Well, it sounds like some of these traits that you've admired in your mentors, this organization you mentioned, I think that is really important for scientists to not only manage their time, but to manage all the variety of different projects going on in the laboratory. Would you consider yourself a particularly organized person?
I am a very organized person. Not that you necessarily notice it if you look at my desk right now, but
it's ordered chaos.
I try to keep on top of email and not go to bed until I've answered every query that I've received for that day. I'm very efficient about writing papers and getting things done that need to get done.
Very cool. Well, can you take us back to the beginning now, Lou, and tell us a little bit about what started your scientific journey? Do you have one of these early memories about when you first started getting interested in science?
I think I was a born scientist. By the time I was 5 years old, I was very much into science. My father did not go to college, but he was in World War II in the Coast Guard and took courses in navigation and weather because you need to know all that in order to be on a ship. And so I remember when I was very young and I would say, why is it raining what makes a rainbow, or the sort of questions that kids ask at that age. Mm-hmm. And most would say, well, because God made it that way, or et cetera, et cetera. My father would go into a great detailed scientific explanation for how the sun warms the water and creates moisture. The moisture gets to a high altitude. It condenses on particles.
Oh, wow.
So by the time I was 6 years old and started school, I knew more science than the science teachers did.
That's awesome. So was he very encouraging of your inquiring mind and asking these kinds of questions?
Oh, yeah. I mean, it was dangerous to ask my dad a question because you could get an hour and a half lecture.
You have to sit down and be prepared for it, I think. So can you tell us what took you then from being this curious kid? What are some of these key steps that brought you to where you are today?
Well, I fell in love with chemistry. Chemistry was my first love. I remember getting a chemistry kit for Christmas one year, and I was mixing all the compounds together, watching things change color, things solidify and melt. And of course, most of the time I made a huge mess out of everything.
Of course.
It was fun. It was like doing magic, but there was a logic to it. So I was fascinated by that. I remember at one time, I probably shouldn't even tell this story, but at one time I wanted to buy firecrackers for 4th of July, but they were illegal in West Virginia. So my father said, well, if you want to make firecrackers, make them yourself. And so I went to the encyclopedia and pulled out gunpowder.
Oh, wow.
To the constituent. I went to the drugstore and bought sulfur, and I shouldn't give you all the recipes, I guess.
Don't worry, I won't make them, but I can't vouch for the listeners.
So I made my own gunpowder and made my own firecrackers, and they actually worked pretty well. Mainly, they just burned really fast.
Right. So were you injury-free after these concoctions?
No, I was actually never got injured from them.
Oh, good.
I decided they worked better as rockets than firecrackers, and so I began trying to make rockets.
That is awesome. So then can you tell us a little bit then about what started your career path in science? So you were interested in science. You were doing these explorations on your own. Did you then start pursuing science in school?
Well, yes. And of course, as I said, I grew up in West Virginia, and I certainly was very interested in chemistry in high school, chemistry and physics and math. My senior year, the physics teacher died the first week of school, and they couldn't find a replacement physics teacher. So the group of us who had signed up to take physics, there were only about 15 of us, I think, decided we would just teach each other physics for the year. So
oh, wow.
There would be a substitute teacher there, but the teacher didn't know any physics. And so we would just read out of the book and discuss it amongst ourselves. I think I learned more physics that year than I could possibly have learned had I had a teacher, because we were forced to teach each other.
Yeah, you have to get a deep understanding and then be responsible for teaching it to your peers. That's really cool. So then tell us a little bit about what happened next when you went to college. How did you decide what major you were going to be, or did you already know that you wanted to major in chemistry?
No, I knew I wanted to major in chemistry. I went to a liberal arts college, West Virginia Wesleyan College, rather than a more technical college, because I wanted also to have a very broad education. And there one was forced to take courses in religion and history and art appreciation and music, et cetera, et cetera. I wanted that broader education, but it also had an outstanding, and still does, outstanding chemistry department. And that was attractive to me. There weren't a whole lot of chemistry majors. And so I got to know all of my science teachers extremely well. But I should say that given what my career has turned into, it seems hard to believe, but I actually never took a biology course in college.
Really?
I really disliked biology in high school because there was so much memorization, and I didn't feel like I was learning any concepts. I was just having to memorize a whole lot of information. And so I swore I would never take another biology course, and I never did. But I reasoned that if you understood chemistry, you could explain all of biology, because biology is just chemistry in action. And that's been true. And in some ways, I think a lot of the breakthroughs I made were totally out of my ignorance, that I didn't understand what everybody was taught in textbooks. And therefore, I just followed my nose. And I discovered something that was different from what was in textbooks. I assumed it was right. And the textbooks were probably wrong.
Well, I love that description of biology as chemistry in action. That's really cool. So then can you tell us what made you decide to pursue the Ph.D., or did you consider at any point an MD PhD?
Well, as I said, if I were to pursue an MD, I would have to take a biology course.
Right. That was the deciding factor.
So no, I didn't pursue that at all. Actually, my option when I went into college, I was planning to go into chemical engineering. And I worked during the summers at Union Carbide, where they were making chemicals and shipping them out. And I had to do quality control assessment on all the things they were doing there. It was extremely valuable experience because I learned so much chemistry at a practical standpoint during those years. But I also realized that I didn't want to go do chemical engineering, because you could pretty much, whatever I needed to know, I could learn within a few months, and the rest of the time would be pretty much repeating things over and over again, where I was much more interested in discovering new things. And that's why I went for the Ph.D. at Cornell.
Certainly. So now I know you have experience working in a variety of different places, and you are also co-founder of some companies as well. So can you talk a little bit about how you've been balancing the academic with the industry side of things?
Well, in academia, we very often make discoveries and unexpected observations, and these can tell us potentially new targets for making drugs, new enzymes which inhibited might be useful for treating a disease. But actually developing a drug is a far more complicated process that takes years. It takes a lot of expertise in chemistry, or medicinal chemistry in particular, which is an art and science in itself. So in order to translate the discoveries that one makes in an academic lab into an actual drug, you almost have to do it with venture capital funding or somehow in an industry setting where people are really trained to do all that work.
Sure.
So that's why I've started several companies over the years. And I love that transition of taking something all the way into a human clinical trial. And it's just in seeing people really benefit from those discoveries that were made. As I say, it's typically 9 years from the time you start trying to make the drug until you actually have a drug in patients or even approved.
Sure. I think there's a lot of waiting in science. And it's interesting to hear about this part of your career that you've sort of expanded into the translation aspect. And I think there are a lot of maybe young scientists out there who are interested in pursuing industry routes. So are there any advice that you would give at this point for people who are maybe considering starting their own company, or any resources that you can recommend for them for how you learned all this information?
Well, I have to say I benefited a lot from my wife having transitioned into industry before I did. She was also an academic. Her name was Vicky Sato. And at the time we got married back in the 80s, we were both on the faculty at Harvard. She decided to start a company, a monoclonal antibody company initially, but then she transitioned into an established biotech company called Biogen, where she became chief scientific officer, and then later moved to Vertex, where she ultimately became the president. So she had gone through the experience of being an immunologist into actually someone who's developing drugs. And watching her complexities of how that happened, because I got to know both of those companies quite well while she was there, it was a real learning experience for me. So when I began starting companies, I really relied a lot on her advice, both in recruiting people into key positions and also in anticipating what the problems are going to be in trying to do drug development. So I had some advantages there.
Definitely. Well, great to hear about some of the adventures along your scientific journey. And I want to talk a little bit more about some of the projects that you have going on right now in the lab that you are just the most excited about. So can you give us some detail?
I'll tell you about maybe 2 stories that I think the listeners probably could understand at some level. One is a very recent observation we've made with vitamin C. As you know, it's an oral vitamin that you get from orange juice in general. Mm-hmm. Fruits have a vitamin C in them. Its technical name is ascorbic acid. And many years ago, back in the 70s and 80s, Linus Pauling, one of the more famous scientists of the 20th century, became obsessed with the idea that vitamin C could cure a lot of diseases, including the common cold and cancer. It wasn't completely clear how he thought it worked or why it would work. But he did end up persuading physicians to run some clinical trials on cancer in which they injected vitamin C at very high doses in the bloodstream of cancer patients and got some remarkable responses. So we were trying to understand how that worked. And I should say later studies with oral vitamin C, where you take pills, didn't really give such convincing results. And we were studying some colorectal cancer cell lines and discovered that the vitamin C could get converted into a different molecule from the reactive oxygen species being produced by the cancer. So lots of cancers are continually converting oxygen into a very reactive species. It's similar to peroxide, like you buy.
Hydrogen peroxide, yeah.
And that peroxide will convert the vitamin ascorbic acid into something called dehydroascorbic acid. And dehydroascorbic acid uniquely can get into cancer cells through a glucose transporter. So it looks a little bit like glucose. And cancer cells are very much addicted to glucose. So they're eating glucose at a very high rate. And this dehydroascorbic acid sneaks into the cancer cell like a Trojan horse through that glucose transporter. And we were able to show that that's how these cells were dying. they were getting killed by this dehydroascorbic acid. So it wasn't really the ascorbic acid that was killing the cancer cell. It was this oxidized form. And in some way, the cancer cell was killing itself by producing the peroxide that was converting that ascorbic acid into dehydro. And that meant there was a very nice efficacy-toxicity ratio. In other words, cancer cells were very susceptible to being killed while normal cells were not. And that is leading us now to do a clinical trial to test this idea that the cancer cells that have these very high rates of glucose uptake and also have high rates of producing these reactive oxygen species like peroxide, that those will be the ones that are most likely to be killed by the IV vitamin C. And we have ways of predicting which patients should behave that way.
Oh, that is really cool. Lou, I think you have brought the chemistry to life, talking about this dehydroascorbic acid and kind of giving it a personality going in like the Trojan horse. So great description. Now, was there another project you wanted to tell us about?
Well, the other thing that I'm working on is insulin. I think everybody knows something about insulin. Type 1 diabetics can't make insulin. Type 2 diabetics have initially what's called insulin resistance, and then they ultimately can't generate enough insulin to function properly, so their glucose levels are out of control. And I discovered back in the 80s an enzyme that mediates everything that insulin does, something called PI3K. So the series of awards that I've received over the years have really been given to me for that discovery. And I discovered that enzyme because it was co-purifying with a variety of what we call oncoproteins. In other words, proteins that are made from oncogenes, cancer genes. And these proteins were shown to drive cancer cell growth. And it turns out that PI3K itself is an oncoprotein. It gets mutated in a vast number of cancers. So this observation that this PI3K is doing two things. It is mediating everything insulin does in your body, but it also, when hyperactivated, causes cancer growth. And that led me to the concept that insulin resistance in type 2 diabetes, where you have very high levels of insulin in your bloodstream, that could be driving the growth of cancer cells. And therefore, that we should be advising people not to eat sugars and rapid release carbohydrates because they make your insulin level go sky high. And if insulin is what's driving a lot of cancers, then you're putting yourself at very high risk for cancer by eating sugar. So I was invited by Sanjay Gupta for an interview on 60 Minutes about the dangers of sugar, in which I explained this, and I've been interviewed on a number of television stations over the years, last 5 or 6 years, specifically on this topic. And the data supporting what I've just said is growing stronger and stronger in our preclinical models and also in human trials that are currently in process. So this is a message that I'm now continually preaching. I started out as a chemist, but I'm telling you,
right.
What we've learned is that sugar is one of the most dangerous things that we can eat. So I never eat sugar. I never eat anything that has sugar added to it. I'll eat fruit occasionally, but I definitely don't eat anything that has sugar added to it.
Now, this is really high impact research. I mean, we talked about the implications not only for cancer, but for things like diabetes. And I'm curious, you mentioned this PI3K is integral to this process. Can you tell us, do you have any suspicions about why this enzyme is particularly susceptible to mutations or what is causing these mutations?
Of course, this very same reaction I mentioned earlier, that cancer cells tend to have a lot of ROS, reactive oxygen species, peroxides, like molecules being generated. So those molecules will cause point mutations in DNA. And of course, most of the point mutations are not necessarily of any benefit to the cancer. But if a point mutation occurs in a gene like PI3K, and it happens to be in the right site, then any cell that has that mutation will have a growth advantage over the surrounding cells. It's as though insulin is telling it to grow all the time, even if there's no insulin there. That actually can happen spontaneously even during embryonic development, where a single cell in the embryo picks up an activating mutation in PI3K, and that every cell downstream of that, every cell in the lineage downstream of that cell will have a growth advantage over every other cell. And the consequence, fortunately, it's a rare disease, but the consequence is that all those cells will now just be growing continuously, and the patient will have maybe an entire leg that's just 3 times bigger than the other leg, or maybe it might only be a thumb or just a toe or something. But it can also happen in the brain, and then you get outgrowths of the brain that are really quite horrible because they result in epileptic seizures. They have to be surgically removed. So this will happen spontaneously at very low rates. But those are not real cancers. In cancers, other things still have to happen in order for the cancer to ultimately become lethal and metastatic. But often it is this ability to grow out of control that is at least one of the steps in the emergence of cancer.
Certainly. Well, Lou, this is fascinating work. And like I said, a huge impact on a lot of different medical conditions, not only cancer, but things like diabetes, obesity. I think there are so many implications for the work that you're doing. So fantastic to hear more about it. But I think we've talked about a lot of the high points in your career, some of the great adventures you've had along your journey. But I want to take a moment to hit some of those low points as well. So we're not sort of giving our listeners a stilted view of what life of a scientist is like. So we want to ensure that people talk about these challenges, these tough times, so that they can help our listeners get through some of these times if they're going through the same thing as well. So can you share a story of your own?
Yes. Well, actually related to the PI3K story, when I made the first observation that this enzyme existed, that it co-purified with a variety of oncoproteins that would cause cancers in chickens and mice, and published that in the mid-1980s. At that time, we didn't fully understand what was going on. But when other laboratories tried to repeat our results, they were not able to repeat them.
Oh, no.
And the reason they couldn't was because we would sonicate our lipids to make them into synthetic membranes. So they had structures very similar to the membranes in cells, while other laboratories would just add detergents and solubilize their lipids. So they were added more like detergent micelles rather than continuous membranes. And the PI3K enzyme requires that its substrate, which is a lipid, be presented in a membrane-like environment. Otherwise, it was totally inactive. And so the other laboratories that tried to repeat our results had no experience in making synthetic membranes, which I'd been doing for many years from the time I was a postdoc with Guido Guidotti. So as a consequence, if they did the assays with detergent, they could not get any activity. And so there were a series of papers written, including by people with Nobel Prizes, saying that they could not repeat our results. That was a difficult time because I also was not given tenure at Harvard at that point, and fortunately got a very good offer to go to Tufts, where I could continue the work. So eventually I had to send my graduate student to these other laboratories with the machines needed to make these synthetic membranes and provide them with the synthetic membranes. And then when they did the experiments, they got exactly the same results we got. But it took about 4 or 5 years for people to generally accept this discovery because it was so unexpected. We went on to show that the enzyme was making a lipid that had never been seen before in nature. So people in the field were saying, well, how is it for 40 years everybody has missed this?
Right.
Many, many labs for 40 years had missed this enzyme. So there was just a general refusal to believe that it could possibly be true. But eventually everybody realized it was true.
Right. You just had to stay the course. So when these contradictory papers came out, did you kind of know exactly why they weren't getting the same results? Or did you have to kind of dig around a little bit to see what might be happening in their laboratories that was not happening in yours?
No, I immediately knew why they were getting the wrong result. I just read the methods and saw that they were adding detergent. And our papers had said you have to do it with sonicated lipids, but these people were not experienced with working with membranes. So.
Sure. So then once this was sort of generally accepted in the literature, was there a moment of catharsis, of relief, once people started to realize that you weren't crazy?
Yeah, but it took a number of years because a lot of people just kind of wrote it off and they were not trained in that field. And so they were afraid to venture into that field. And so it wasn't immediately accepted. It took several years.
Certainly. Well, Lou, thank you so much for sharing one of these tough times of your own. I can imagine that is a difficult position to be in, and it's great to hear that you made it through. But we don't just want to talk about these tough times. Lou, I want you to share with us next a success story of yours that was a big discovery or even a little moment that just meant a lot to you.
Quite honestly, the discovery of PI3K, and I should say that the way that even before I knew completely what was going on, the day that it told me that we'd really made a huge leap would sound like a rather trivial observation, that my student was comparing the products of the PI3K, which at that point we didn't call PI3K, we called it type 1 PIK, because we didn't know that it was phosphorylating the 3 position. The 3 in this means that it's phosphorylating the 3 position of the inositol ring on this phosphatidylinositol lipid. And at that time, the only known lipid that had a single phosphate on it was PI4P rather than PI3P. And so we had PI4P as a standard, and we were running the PI3P on a thin layer plate to do chromatography. And we noticed that there was a 1 millimeter difference in the position where the two migrated.
Wow, 1 millimeter. I don't think I would have noticed.
Yeah, if you ran the two together, they would look like they were right on top of each other. But because we had every other lane was PI4P, PI3P, 4P, 3P, 4P, 3P. And that said to me without any doubt, there was a reproducible 1 millimeter difference in the migration that chemically had to be a different molecule and therefore was not PI4P, but was something different. I went home and opened a bottle of champagne and I said, now I know why this pathway is completely different than everything everyone else has been working on. It's actually a completely different molecule.
That is awesome. So you had a toast to the PI3K?
Yeah. Actually, that evening, I didn't know it was PI3K. I just knew it was not PI4K.
Hey, well, that is the start of an exciting journey there. So great to hear about one of these successes. You mentioned you celebrated with a bottle of champagne. Was this something that you discovered fairly independently, or were there a lot of people involved in this discovery?
Well, it was my laboratory that made discovery. Now, in fact, I collaborated with a number of laboratories to purify the enzyme, to characterize its effect on cells. Was work particularly with Tom Roberts' laboratory and Brian Schaffhausen's laboratory. The actual observation that it was making an unexpected lipid was from work in my laboratory with Malcolm Whitman, a graduate student who was really running that project.
Well, fantastic to hear about one of these successes from your laboratory. And I think there have been so many exciting directions from this success. So we've talked a little bit about the different aspects of a science career that are fantastic, your discoveries, your journey to get to where you are today, some of these exciting moments of success. And I think another wonderful aspect of the career in science that we haven't yet really touched on are some of the opportunities that you get to travel, to see the world. So Lou, can you think back on all the places you've been for science and tell us about your favorite experience and where you were?
Yeah, I certainly travel quite a bit to meetings. In fact, I just got back from Chile last week in the high plains, sort of at the foothills of the volcanoes in the Andes, where it hasn't rained in about 50 years. It's like the driest place on earth.
Oh, wow.
But fantastic scenery there. There are a number of meetings that I've gone to that were sponsored by an organization called the Ipsen Foundation that's funded by a group in Paris. It was a foundation set up by the Ipsen pharmaceutical company to host meetings in various disease areas. And about 12 years ago, I guess it was, they decided to host a series of meetings on cancer. And there's each year about 30 or so scientists are brought in. There's a core group of us who almost always go. And then there's another 15 or so who are brought in as specialists in a particular area of cancer that we're going to talk about at that meeting. The very first one of these I went to was one of the more exotic meetings I'd ever been to. The meeting took place in Agra, India, which is where the Taj Mahal is. It was my first trip to India, and so flying into New Delhi and then taking a bus from New Delhi to Agra was the most unworldly experience.
That's a good way to describe it.
In that every possible mode of transportation was on that road. Carts were being pulled by dogs and goats and camels and elephants and people with bicycles, and just every possible mode of transportation was going on, all in competition for the same roads. And of course, water buffaloes would sit down in the middle of the road and everybody would have to stop and go into the opposite lanes.
Right. Honk a little bit. And
then the contrast to the Taj Mahal, which is the most spectacular piece of architecture I've ever seen. It was definitely not a disappointment to go there. And Agra Fort, which is also in Agra, also a beautiful building. The poverty of many of the people living in that area is just such a contrast. But on top of that, of course, we had a fantastic scientific meeting where we got to share ideas openly and lots of discussion. It was really set a standard for scientific meetings, in my opinion.
Well, I think it's tough to have one of your first meetings be one of these exceptional experiences, what you measure everything up against next. Very cool to hear about your experience in India. And I think one of the wonderful things about traveling is you get to see the different ways that people live and get to talk to scientists from all over the world. So have you been able to form this group of colleagues, this network of people across the world that you've kept in touch with?
Yes, I know all those people quite well, and many of them we get together at least once a year at this type of meeting. I should add that typically on these Ipsen meetings, we also invite scientists locally from the country that's hosting us. So we get a chance to learn a bit about the local diseases and what they're dealing with. This was one of the first times I learned about the high incidence of type 2 diabetes in India. Look at these people who are very, very thin, and yet as they're going on Western diets, they develop insulin resistance at extremely high rate. Their bodies are not accustomed to eating sugar and rapid release carbohydrates, and it all gets converted into fat in the liver and then ends up causing them huge disease problems, including increasing their risks of cancer. So that meeting also gave me some more information to think about with regard to these connections.
Certainly, and brings a new perspective to how you're thinking about your research. So wonderful to hear about these travel experiences. And you sort of hinted a little bit at one of the great things about the experiences in science are some of the people that you get to work with and the people you get to collaborate with. And I love talking to scientists about the fun and fond memories that they've shared with their colleagues. And I think there are so many different personalities. There's so many fun, quirky, awesome people in science. But I think that's not always what the stereotypes in the media reflect about scientists. So we like to dispel some of those stereotypes by sharing fun stories. So can you share one of your own experiences today, Lou, that highlights the human side of science?
Well, my laboratory has always, I guess it's my own personality, but I've always attracted really crazy people into my lab. Crazy smart. And I like to think that for the most part, we get along extremely well and people share things and share ideas. They're very open. And yet many scientists go into science because they are so driven by the science that they're not particularly social. So part of my goal is to get people to open up and talk to each other more. And breaking those barriers requires that you get them out of the lab every now and then and into a setting where they're forced to talk to each other. So I have a retreat each year in which we spend 2 nights and 3 days. And every single person in my lab has to give a talk. All the graduate students and postdocs give a 15-minute talk each. But I also invite another 20 or so ex-postdocs or graduate students, and they also give talks. So that's why it takes 3 days to get through all of this.
Gotcha.
But we have a lot of fun. So we generally go on these retreats in pretty interesting locations like a beach. And one night the women are required to fix the food, and the next night the men are required to fix the food. And we have sort of an ongoing competition as to who makes the better meal. And then after we finish dinner, then we start dancing. So music and dancing that goes on until about 3 or 4 in the morning.
Oh, wow.
And finding a location where the police don't arrive has always been a challenge. But we found a few locations. We have to be like out on the beach in the very last building away from the rest of the hotel.
Right, where you won't disturb everybody.
One year we actually decided, one of my technicians decided it would actually be cheaper for us to go on a cruise for our lab retreat. So they had a 3-day cruise. So we did that. And fortunately, they had a dance floor in the ship. So
good thing. So are you a good dancer?
Oh, yeah, I love dancing. I've always enjoyed dancing. My daughter is actually a professional ballroom dancer.
Oh, very cool. So do you do ballroom dancing primarily?
I can do a little ballroom. I'm more disco than ballroom.
Gotcha.
Similar moves.
Sounds like you've got a wide repertoire. Now, Lou, you mentioned that you have a sort of cooking contest that happens in these retreats. So are you much of a cook as well?
No, I'm not a cook at all.
More judging and guiding on the sidelines?
We made an agreement when my wife and I got married that because she's a fantastic cook. Oh, by the way, she's also a terrific ballroom dancer.
Wonderful.
But she's a terrific cook and loves cooking. And so we made agreement that she cooks and I clean up. And it's really a bad agreement because she has no incentive to use a minimum number of utensils.
Right. No reason to make your job easy, of course. Well, these lab outings sound phenomenal. It sounds like you guys have a great time on these retreats. And I can't imagine 3 days of listening to talks and kind of getting to hang out with some of the previous postdocs and graduate students in your lab. That must be wonderful for your current students.
It also teaches people to be able to go without any sleep at all and still be able to perform.
Right. And then they're back in the lab the next day, right? Back to work. Well, Lou, great to hear about these traditions. And I think these traditions are really important for making the lab a fun environment and for kind of showing people that you can work hard, but you can also have fun too in science. So we've talked about a lot of different aspects of your career, and we've talked about some of your successes and the wonderful things you've been able to do. And I want to hear, is there a particular piece of advice when you think back over all the people who've helped you? Is there one piece of advice that rises to the top in terms of what was the best advice you've ever received?
I think my father gave me the best advice, and my mother as well. They were both very confident that I could do anything that I wanted to do. My father, first of all, he was famous for saying, nothing is impossible. And secondly, he gave me the confidence that there was nothing that I couldn't figure out how to do. And as I say, he was teaching me science at a very early stage, and he would give me responsibilities. We built a house when I was 15 years old. Every single aspect of the house, the plumbing, the wiring, the roofing, everything my father and I did with some help from my cousins and brothers. So he just instilled within me the confidence that there was nothing that I couldn't figure out how to do.
Well, that is very impressive, building a house from scratch. I like it, Lou. And now you mentioned this idea of being able to do anything you want to do. And I want to ask you one of these big questions where you can do anything you want to do. So can you tell me, Lou, if things like funding and staff and technology and feasibility, none of these barriers in science were a problem, what is the one question that you would want to address?
What I would do, and this requires more than just money, it also requires buy-in from many people. But I think if we want to cure cancer, we have a full, clear path of how to do it. We need every single patient who comes down with cancer to agree to allow the mutational information, in other words, what happened to drive their cancer, to be shared in what we call a de-identified way so that nobody would know whose data was whose. But that data, detailed sequencing of the entire genome of their tumor, along with the clinical responses that they had to whatever drug they were treated, if we could get that information shared across everybody in the United States, or even better, everybody in the world, and then in a real live time be able to get onto a computer, look at that information, see it changing on a minute-by-minute basis as people go on new drugs or new tumors sequenced and dumped into the database. And then when you see people respond, you share that information. These set of patients took this drug, they had pancreatic cancer, and their tumor shrunk. And therefore, if the next patient who comes along has that same set of mutations, that's the drug they should take. And it would be remarkable how fast we could accelerate the cures for cancer if it were possible to share that kind of information. It requires massive computing, but then we have massive computing already in systems like Google. But it does require that all the physicians agree and the patients agree that this kind of information should be shared.
Well, Lou, I think this mega cancer database you're talking about is a brilliant idea. So do you know, are there efforts currently going on moving us in that direction?
Certainly plenty of discussions going on. I think Obama's announcement about the moonshot, that's certainly one of the things that is being discussed with regard to how we go forward.
Well, exciting things for us to look forward to in the future. Lou, thank you for dreaming big with us, and thank you for chatting with us. Now, is there any other last piece of advice or maybe a last note of inspiration that you want to leave our listeners with today?
I always tell everyone to do whatever is easiest for them to do. It's counterintuitive that you should go do something really hard. The truth is, whatever is really easy for you to do, that's your talent. And there's a way, almost always, if you pursue your talent, you can find a way to make it useful to people and to yourself. So follow your talent.
Phenomenal advice, Lou. I like it. Can you tell our listeners now where they can go if they want to learn more about you and your fantastic research?
I guess I do have actually, all you have to do is Google my name. There are plenty of connections out there.
Absolutely. There's some fantastic videos out there as well. And I think the 60-minute video that you mentioned is up on the internet as well. So Lou, thank you so much for chatting with us today. It's been a pleasure to have you on the show.
Thank you.
All right. And listeners, thank you for joining us. We'll see you next time on another episode of People Behind the Science. Your voyage to explore the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to chatting with you next time on People Behind the Science.